Welcome to the Deep Dive, where we take complex information, break it down, and really try to uncover the most fascinating insights for you.
And today we're diving deep, really deep into the foundational elements of life itself, amino acids and their absolutely critical role in building proteins. We're going to be working through a key chapter from Marks Basic Medical Biochemistry, focusing on how these tiny sort of versatile molecules build the complex machinery of our bodies.
Yeah, our mission today is to transform these, let's face it, sometimes dense biochemical concepts into clear, engaging takeaways. We want you to understand not just what amino acids are, but why their unique properties are so incredibly crucial for, everything from carrying oxygen in your blood to helping doctors diagnose a heart attack. Get ready for some aha moments, definitely. Okay, let's start right at the very beginning. When we talk about proteins, we're talking about these, ultimate workhorses in your body, aren't we?
Oh, absolutely. You got it. Proteins are truly the ultimate multitaskers. They're transporters, kind of like tiny delivery trucks carrying compounds in the blood. They act as cell adhesion molecules, holding cells together. They function as hormones, carrying vital signals. They form ion channels that let things pass through membranes. And of course, they're the enzymes that speed up pretty much every biochemical reaction you can think of. They really do it all.
So, okay, if proteins are these incredibly versatile machines, what is it that makes each one unique? What's the secret sauce?
Well, it all boils down to its linear sequence of amino acids. We call that its primary structure. This specific, unique order dictates, well, everything about that protein. How it folds into its 3D shape, how it interacts with other molecules, everything.
Thinking about it, it sounds like proteins are built sort of like a string of very specific beads. Is that a fair comparison?
That's a great analogy, yeah. Imagine a string of, say, 20 different types of beads. Those are our amino acids. All human proteins are built from just these 20 basic beads. And what's really fascinating is that the order of these beads is determined by your genetic code. That's a sequence of 3 DNA bases, or nucleotides. So each gene, then, is essentially a recipe written in DNA that codes for a functional product, usually a protein chain.
Right. So if the genetic code is the blueprint, what happens if there's even a tiny error, a mutation in that blueprint?
Yeah.
How big can the impact of such a small change actually be?
Oh, the impact can be absolutely profound. A classic and pretty stark example is sickle cell anemia. This inherited disease is caused by just a single nucleotide mutation in the gene for one of the hemoglobin subunits.
Hemoglobin, right? That's the critical protein in our red blood cells. It's the one responsible for transporting oxygen all around the body.
Exactly. And adult hemoglobin is actually quite complex. It's made of 4 long chains of amino acids, what we call polypeptide chains, 2 alpha chains, and 2 beta chains. Now, in sickle cell anemia, that single mutation changes just 1 amino acid in those beta chains. A negatively charged glutamic acid gets replaced by a hydrophobic valine. This tiny swap, just 1 amino acid out of hundreds, has truly catastrophic effects on how the protein functions.
Wow. It's incredible how one tiny change can ripple through a whole system like that. Let's zoom in on these amino acid building blocks themselves. What do all 20 of them have in common? What's their basic structure?
Okay, so every single amino acid shares a common core structure. There's a central carbon atom, which we call the I carbon. Attached to this I carbon are 4 key things: a carboxylic acid group, an amino group, and this is always in a specific L configuration, which is crucial for how our bodies use them, a hydrogen atom, and then the really important part, a unique side chain. That's also called an R group. It's the side chain that makes each amino acid distinct, gives it its own personality, if you like.
And when they're just floating around in solution, maybe in your blood or something, do they carry an electrical charge?
They absolutely do. At physiological pH, so the normal pH in our bodies, they exist as what we call zwitterions. Basically, this means the amino group picks up a positive charge, and the carboxylate group carries a negative charge. So it's got both. This dual charge actually makes them highly water-soluble, which is essential for them to move around and do their jobs in our, watery biological environments.
Okay, so when these individual amino acids link up to form those long protein chains we talked about, how do they actually connect? What's the bond?
They connect through what's called a peptide bond. It's a specific type of covalent bond. This reaction happens between the carboxylic acid group of one amino acid and the amino group of the next one. Imagine them forming that long, continuous bead string again. The amino group, I carbon, and carboxyl groups form this repeating peptide backbone. And it's from this backbone that those unique side chains stick out, giving the protein its specific character. These side chains, They really are what make all the difference. They dictate folding and function.
Right, so those side chains are absolutely key to a protein's characteristics. When biochemists look at these 20 different amino acids, how do they usually categorize them? How do they make sense of all that diversity?
Well, we often group them based on the polarity of their side chain. Basically, whether they're charged, nonpolar and water-hating, or uncharged but still polar. Or sometimes we group them by structural features like being aliphatic, straight chains, cyclic rings, or aromatic special rings. Thinking about these classifications isn't just about labels. It's like understanding the fundamental grammar that dictates how a protein will fold, what it'll bind to. It even influences whether it'll be part of a cell's outer layer or maybe its internal engine. Understanding these groups gives you a kind of predictive map for, well, nearly every biological process.
And I've heard about something called a hydropathic index. What's that telling us?
Ah, yes, the hydropathic index. It's basically a numerical scale that tells us how much a side chain dislikes water, how hydrophobic it is. So a more positive index means it's more hydrophobic. These amino acids will tend to cluster together inside a protein trying to get away from water. You'll often find them tucked away inside the core of folded proteins or maybe embedded within cell membranes. On the other hand, a more negative index means the side chain is more hydrophilic water-loving. So you'll typically find those on the protein surface interacting happily with the surrounding water.
Okay, let's unpack some of these categories. Maybe give us a few examples. What about the nonpolar aliphatic ones?
Sure. The simplest one is glycine. Its side chain is just a hydrogen atom, tiny. Because it's so small, it has minimal steric hindrance, it doesn't bump into other atoms much. So glycine is often found in really tight bends or turns in proteins, allowing for very compact, flexible structures. Then you have others like alanine, valine, leucine, and isoleucine. These all have bulkier, nonpolar side chains, very hydrophobic. In proteins, they're the ones that cluster together in the core, forming those water-hating pockets. These pockets are stabilized by weak forces called van der Waals forces.
Proline sounds a bit different in this group, though, like an odd one out.
Proline is definitely unique. Its side chain actually loops back and forms a rigid ring that involves its own I-carbon and amino group parts of the peptide backbone itself. This rigid cyclic structure introduces a distinct kink or bend in the polypeptide chain. It restricts the protein's flexibility right at that point. It's almost like a structural brace that locks a certain conformation into place. Quite important for structure.
Okay, so we've covered the simpler sort of straight-chain amino acids, but what happens when you introduce more complex shapes, like ring structures? Tell us about the aromatic amino acids.
Right, the aromatics, these all contain characteristic ring structures. Phenylalanine is very nonpolar, very hydrophobic. Then there's tyrosine. It has a hydroxyl group OH on its ring, which makes it more polar. It can actually form hydrogen bonds. And tryptophan has an even more complex structure, an indole ring with a nitrogen atom, also making it quite polar compared to phenylalanine. Oh, and an interesting side note. These aromatic rings absorb UV light, which is a property biochemists use all the time to measure how much protein is in a solution.
Interesting. Okay, next up, the polar uncharged amino acids.
Right. These include asparagine and glutamine, which both have amide groups in their side chains, and also serine and threonine, which have hydroxyl groups, sulfonyl OH. The key thing here is that their side chains are great at forming hydrogen bonds. Because they like interacting with water, you'll typically find them on the surface of water-soluble proteins interacting with the surrounding aqueous environment.
Makes sense. And what about these sulfur-containing amino acids?
Ah, yes. Two important ones here. First, cysteine. It has a sulfhydryl group, -H. And what's fascinating about cysteine is that two of them can actually form a covalent bond between their sulfur atoms. This forms a disulfide bond, creating a linked unit called cysteine. These disulfide bonds act like internal staples, really important for holding protein chains together or stabilizing specific regions within a single chain. They add a lot of structural integrity. Then there's methionine. It also contains sulfur, but its side chain is nonpolar and it cannot form disulfide bonds. Its main role is often in initiating protein synthesis or transferring methyl groups in certain reactions.
Okay, finally we get to the charged amino acids.
Yeah.
These must really change the game for how proteins function, right? Big impact.
Oh, absolutely. These are definitely the heavy hitters when it comes to electrical charge. We have aspartate and glutamate, which are the acidic ones. They carry a negative charge at physiological pH. And on the basic side, we have histidine, lysine, and arginine. These all carry a positive charge. Their charges allow them to form strong ionic bonds. You might also hear them called electrostatic bonds or salt bridges with oppositely charged groups. These interactions are vital for protein stability, enzyme activity, binding other molecules. Really crucial stuff.
So these charged side chains can significantly impact how a protein interacts with other molecules, and even how its behavior changes depending on the environment, like the pH.
Precisely. And understanding these charge dynamics, how amino acids behave at different pH levels, isn't just theory. It leads us to concepts like pKa and the isoelectric point, or pi. These properties tell us exactly how the charge on an amino acid or even a whole protein changes as the pH changes. This is super important for practical things, like how a diagnostic test like electrophoresis works. It separates proteins based on their net charge, helping identify proteins with different amino acid compositions. It also helps understand how a drug might bind differently in an acidic stomach versus neutral blood. And it explains things like in the case study of David K. with cystinuria. Remember him? His problem with amino acid transport led to painful kidney stones. His defective transport proteins couldn't reabsorb certain charged amino acids, cysteine, arginine, lysine, in his kidneys. So the less soluble cysteine, formed from two cysteines, precipitated out and formed those painful stones. It all comes back to charge and solubility.
Wow. Yeah. It's clear how powerful even a single amino acid change can be, like we saw with sickle cell anemia earlier. But thinking more broadly, the primary structure isn't always identical from person to person, is it? Or even within the same person.
That's a really crucial point. No, the primary structure can vary quite a bit. You'll find variations within the human species, between different tissues in the same individual, or even at different stages of development. Often these variations are tolerated just fine, especially if they occur in non-critical regions of the protein parts that aren't essential for its core function, or if they're conservative substitutions. That means replacing an amino acid with another one that has very similar properties, like similar size and polarity. The protein can often still function normally.
But what happens if a change hits a really critical region, a part that's absolutely essential?
Then it can definitely lead to dysfunction, sometimes severe. This happens with non-conservative substitutions, swapping an amino acid for one with very different properties, like size or charge, especially if it happens in an invariant region. An invariant region is a part of the protein sequence that's remained unchanged across different species or variations because it's absolutely essential for function. The sickle cell mutation we talked about, that's a prime example of a non-conservative substitution in a critical spot, swapping that charged glutamate for a hydrophobic valine.
Right. And you mentioned sickle cell is inherited. Thinking about someone like Will Esser from the clinical examples, what's his specific genetic situation?
Okay, so Will S. is described as homozygous for the sickle variant of the globin gene. That means he inherited two identical copies of this altered gene, one from his mother and one from his father. This leads to that specific substitution we discussed, valine replacing glutamate at the sixth position of the globin chain. And when Will's red blood cells are deoxygenated, like in tissues with low oxygen, this new valine creates abnormal hydrophobic patches on the hemoglobin surface. These patches cause the molecules to stick together, polymerize into long rods, and distort the red blood cells into that characteristic sickle shape.
Okay. You also mentioned the term polymorphism. How does that fit into this picture of variation?
Right. Polymorphisms are basically variants of a gene or allele that occur with a significant frequency within a population. Sometimes they cause disease, sometimes not. The sickle cell allele is actually a very famous example of a polymorphism. And here's where it gets really interesting from an evolutionary perspective. Individuals who are heterozygous for the sickle cell trait, meaning they have one normal vt-globin gene and one sickle cell allele, actually have a survival advantage in regions where malaria is common.
How does that work?
Well, the malaria parasite lives inside red blood cells. In heterozygotes, the red blood cells containing the parasite are more likely to sickle and get destroyed by the spleen. This helps eliminate the parasite before it can cause severe disease, so they get some protection. But for homozygous individuals like Will, who have two copies of the sickle allele, the sickling is much more severe and happens more frequently. This leads to serious health problems like those vaso-occlusive crises we mentioned.
Ah, okay. So the sickle cells clog up tiny capillaries.
Exactly, causing intense pain and damage to tissues and organs. It's a really tough condition.
That's a fascinating, though harsh, example of selective pressure. Okay, moving beyond individual variations, proteins can also be grouped into families, right? Based on evolution.
Absolutely. We talk about protein families and even larger superfamilies. These are groups of proteins that have evolved from a common ancestral protein, often through processes like gene duplication followed by divergent evolution. Basically, nature copies a useful gene, and then the copies gradually change over time, allowing the resulting proteins to take on new but often related functions.
Like myoglobin and hemoglobin, perhaps.
Precisely. They're textbook examples of paralogs. That means they're related proteins found in the same species, but they perform different functions. Myoglobin is a single polypeptide chain. Its main job is storing and transporting oxygen within muscle cells, kind of like a local reserve tank. Hemoglobin, as we know, has 4 chains and is specialized for transporting large amounts of oxygen from the lungs to all the body's tissues via the bloodstream. Despite these functional differences, they share many structural similarities and conservative amino acid substitutions, clearly showing their common evolutionary origin.
And proteins can also vary depending on, say, the stage of development or the specific tissue they're found in.
Yes, absolutely. We see striking developmental variations, especially with hemoglobin. For instance, fetal hemoglobin, HbF, which has alpha and gamma chains, has a much higher affinity for oxygen than adult hemoglobin, HbA, which has alpha and beta chains. This higher affinity is a crucial physiological adaptation for the fetus. It allows the fetus to effectively pull oxygen from the mother's bloodstream across the placenta, even in that relatively low oxygen environment.
Makes sense. And what about tissue-specific isoforms, or sometimes called isozymes?
Right. A great example there is creatine kinase, or CK. This enzyme exists as different forms, or isozymes, in different tissues. They all catalyze the same reaction, converting creatine to phosphocreatine, an energy storage molecule, but they have slightly different amino acid sequences in the structures. For example, skeletal muscle primarily makes a CKMM isozyme, the brain mainly produces CKBB, and the heart muscle produces both MM and BB, and also forms a hybrid dimer called CKMB.
And these differences are diagnostically important in medicine, aren't they? You can tell where damage might have occurred.
Exactly. They're very useful clinically. In the case study of Dianne J, who had a myocardial infarction, a heart attack damage to her heart muscle caused enzymes to leak into her bloodstream. Measuring elevated levels of the heart-specific CKMB isozyme in her blood was a way to confirm heart muscle damage. Nowadays, we often use even more specific markers like cardiac troponin T, CTNT, but CKMB was historically vital and is still a valuable tool for diagnosis.
Okay, and one last area of variation between different species. This has had a huge impact on medicine, especially with things like hormones.
Indeed. A classic example is insulin. Insulin, as you know, is the hormone essential for regulating blood glucose levels. Historically, before we could synthesize human insulin, diabetes was treated using insulin purified from cows, beef, or pigs, pork. Now, insulin is highly conserved across species, meaning the sequence is very similar. However, there are minor amino acid differences in what we call variable regions. For some patients, these small differences were enough to trigger an immune response against the animal insulin.
Right, which I imagine spurred the development of synthetic human insulin using biotechnology.
Precisely. Using recombinant DNA techniques, we can now produce large quantities of identical human insulin like humulin. And we've even gone a step further, creating bioengineered insulin analogs like lispro. Lispro is particularly interesting. Scientists made a simple switch of 2 amino acids, lysine and proline, near the end of its B chain. This small change prevents the lispro insulin molecules from clumping together and binding zinc to form larger hexamers.
And why is preventing that hexamer formation important for patients?
Well, normal insulin naturally forms these hexamers, and when injected, these clumps have to break down before the insulin can be absorbed into the bloodstream. This takes time. Lispro, because it doesn't form hexamers, is absorbed much faster. This allows patients like Dianne A in the case study to inject it just minutes before eating rather than 30-60 minutes before. This gives them much more flexibility and better control over their blood glucose levels, helping prevent those dangerous post-meal spikes. It's a fantastic example of how understanding amino acid properties leads to better therapies.
Okay, so we've talked a lot about the primary sequence. But a protein's journey isn't always finished once that amino acid chain is built, is it? There's this whole other layer of complexity, post-translational modifications.
Ah, yes. That's where things get even more intricate and highly regulated. These are chemical modifications that happen after the protein has been synthesized by the ribosome, often even after it started to fold. These changes are usually catalyzed by specific enzymes. They can involve adding various chemical groups, oxidizing certain amino acids, and they can dramatically alter the protein's function, its stability, where it goes in the cell, or how it interacts with other molecules. Think of them as crucial fine-tuning steps.
Could you give us some examples of these important modifications?
Sure. A really common one is glycosylation. That's the addition of carbohydrate chains, or sugars. It happens in 2 main ways. There's O-glycosylation, where sugars attach to the hydroxyl group of serine or 309 residues. And there's N-glycosylation, where they attach to the nitrogen atom in the side chain of asparagine. These sugar modifications are incredibly important, especially for proteins on the cell surface or proteins that get secreted from the cell. They can affect protein folding, stability, protect them from degradation, and act as signals for sorting and targeting.
What about modifications that help proteins stick to cell membranes?
Good question. For membrane association, we often see fatty acylation or prenylation. This involves covalently attaching lipid groups, essentially fatty molecules, to the protein. For instance, lipid groups like palmitoyl, a 16-carbon chain, or myristoyl, 14 carbons, can be attached, acting like greasy anchors that embed the protein into the lipid bilayer of the membrane. Prenylation involves adding specific types of branched lipid groups, like farnesyl or geranyl-geranyl groups, usually to cysteine residues near the protein's end. This is another common way to tether proteins, especially signaling proteins, to membranes.
And there are also modifications that act more like switches, right? Turning protein activity on or off.
Exactly. Phosphorylation is probably the most widespread and important regulatory modification. It's a huge deal in cell signaling. Protein kinases are enzymes that add a phosphate group, which is bulky, and carries a significant negative charge to the hydroxyl group of specific serine, threnone, or tyrosine residues. This addition can drastically change the protein's shape, its conformation, and its activity, effectively acting as a molecular switch to turn cellular processes on or off in response to signals. It's incredibly dynamic.
What about some others like acetylation or ADP-ribosylation?
Yep, those are important too. Acetylation, which is adding an acetyl group, often happens on lysine residues of histone proteins, the proteins that package DNA. This modification changes how tightly the histones bind to DNA, playing a key role in regulating gene expression. ADP-ribosylation involves transferring a whole ADP-ribose unit from a molecule called NAD+ onto specific amino acid residues like arginine, glutamine, or cysteine. This can also regulate protein activity. And interestingly, several bacterial toxins, like the ones that cause cholera and pertussis, whooping cough, are actually enzymes that perform ADP-ribosylation. They hijack host cell proteins with this modification, disrupting normal cell function and causing disease.
Nasty. Are there other modifications tailored for very specific functions?
Yes, quite a few. For example, carboxylation. This adds an extra carboxyl group to certain glutamate residues on blood-clotting proteins. This modification allows these proteins to bind calcium ions, which is essential for them to stick to phospholipid surfaces at the site of injury and initiate the clotting cascade. Another one is hydroxylation, adding a hydroxyl OH group. This is critical for the protein collagen, the main structural protein in our connective tissues. Specific proline residues in collagen get hydroxylated. These added hydroxyl groups allow for more hydrogen bonding between collagen chains, which is crucial for stabilizing the triple helix structure and giving collagen its incredible tensile strength. Without it, you get conditions like scurvy.
Right. And finally, you mentioned selenocysteine earlier. That one's incorporated differently, isn't it? Not a post-synthesis change.
Exactly. Selenocysteine is quite unusual. It's not technically a post-translational modification in the same way as the others. Instead, a selenium atom replaces the oxygen in the hydroxyl group of a serine residue. But this happens while the serine is still attached to its specific transfer RNA, tRNA, molecule before it even gets added to the growing protein chain. Then, this modified sirtRNA, now carrying selenocysteine, is recognized by the ribosome and inserted directly into the protein during synthesis at specific codons. So it's a direct incorporation specified by the genetic code in a complex way, not an after-the-fact modification. That's why it's sometimes called the 21st amino acid. So as we've kind of journeyed through this deep dive, we've really seen how the incredible diversity packed into just those 20 standard amino acids, combined with the precision of the genetic code dictating their sequence, and then layered with all these dynamic post-translational modifications. How all that creates a simply vast array of proteins that perform pretty much every function imaginable in our bodies.
Yeah, it's mind-boggling, from that subtle shift in charge causing sickle cell disease, all the way to the clever engineering of insulin for faster absorption. The story of amino acids really is the story of life itself, isn't it? Dictating structure, function, health, the whole shebang.
It really is. We've navigated the different ways we classify them. We've understood the often dramatic impact of mutations and hopefully appreciated how variations in protein structure, whether they occur during development in different tissues or between species, drive adaptation and also fuel medical innovation.
And thinking about the tools available now, like diving into huge databases like GenBank for DNA sequences or the RCSB protein databank for 3D structures, it just highlights how much more there's still to discover, right? Connecting those sequences to functions and potential diseases, it feels like we're still just scratching the surface in some ways.
Absolutely. And this detailed understanding of amino acid chemistry, protein structure, how they vary, how they're modified, it isn't just, abstract academic knowledge. It's directly applicable. It informs clinical diagnosis, drives the development of new treatments, and fundamentally shapes our understanding of human health and disease.
Okay, so here's a final, maybe provocative thought for you to chew on as we wrap up. We've seen how a single amino acid change can cause a devastating disease like sickle cell. But another single change, or maybe two, can drastically improve a drug like lispro insulin. Looking ahead, how might a future where we can precisely predict and maybe even engineer these molecular interactions at the amino acid level? How might that fundamentally change our entire approach to health and disease? Could we move beyond just treating symptoms to actually curing or even optimizing at the molecular level?
That's the million-dollar question, isn't it? It's certainly the direction things are heading. Thank you for joining us on this deep dive into the really fascinating, intricate world of amino acids and proteins.
Yes, thank you. We hope you feel a little more well-informed, maybe a little more curious about these fundamental building blocks of life. We'll catch you on the next deep dive.